Hello everyone, and welcome to session 5 of the NLM Curation at Scale workshop towards automated curation of model organisms. As most of you know, over many decades, model organisms have contributed tremendously to our knowledge of basic biology, and owing to genome conservation, have informed many aspects of human biology and disease etiology. There is also a long-standing tradition of data curation and dissemination within the model organism communities, practice that has been facilitated by their respective knowledge bases, some of which are now being actively integrated into a common data portal at the Alliance of Genome Resources. So we're fortunate today to have representatives from 4 model organism knowledge bases who will share with us the lessons they've learned about scaling curation and the vision they have for moving the field forward. So just as a reminder, if you have questions, please post them in the Q&A addressed to the relevant speaker. So to start, our first speaker is Dr. Paul Sternberg, the Bren Professor of Biology in the Division of Biology and Biological Engineering at the California Institute of Technology. Dr. Sternberg is primarily a C. elegans researcher and knowledge base user with broad biological research interests. He co-founded Wormbase, the Alliance of Genome Resources, Texpresso, and Micropublication Biology, and has been into curation at scale for 20 years. And so today he will tell us about getting to the future of biocuration.
Well, thanks a lot, Kimberly, and thank you very much to the organizers for inviting me to speak, and really for a fantastic meeting so far. I look forward to the rest of it. Can you see my screen? Is it sharing on? Okay. So yeah, I wanted to try to look and be slightly forward thinking. As Kimberly mentioned, I've been doing this for, say, a couple of decades, and there's been great progress, but I have to say things are still moving a bit slow for my tastes, and I think for the needs of our research communities. So what I'm going to talk about some efforts that have been funded by the National Library of Medicine, of course, the National Human Genome Research Institute, and some from the initial BI. But I want to say that overall, what I'm going to try to argue is that the way we scale is, well, certainly more expert curators will help. Software is really important, as we've been hearing, and this includes artificial intelligence in various guises, and also user interfaces, I think, are really important. It's been touched on this morning. But I also want to stress, making better use of authors and journals, and how we might envision that happening to really bring all these together in a useful way. Okay, so I'm involved in a number of projects, as Kimberly mentioned. Wormbase is the C. elegans information knowledge base. I've been involved in the Gene Ontology Consortium for a number of years. We've been working, and you'll hear from some of my colleagues at the Alliance of Genome Resources, how we can work together to make things go faster and more accurate and more fun to use. When we started Wormbase 20 years ago, I immediately thought, I don't want to read these papers by hand. There must be a way to do it with computationally. I talked to a number of CS and EE colleagues who said it was impossible. And luckily, one person said that his company was working on it, so I knew that it was probably worth doing it. So then Emer Kenny and Michael Mueller and I developed Texpresso, which has been used in various guises and mentioned today. If you went to the poster session, you heard Kimberly discuss the Acknowledge system, which I'll discuss briefly in a few minutes, about how you can use artificial intelligence to empower and take advantage and leverage real intelligence, which is a way to scale expert curation, and also talk about how if we really have control of scholarly communication by biocurators, we could do things even more efficiently. Okay, so let's step back and look at the current data lifecycle. Researchers, like people in my laboratory and many thousands of laboratories around the world, do experiments, collect data, get omics data, individual handcrafted experiments, and they then when it comes time to write a paper or papers, they interpret and integrate that information, and they write the paper and come up with this article. Some of the information is discarded because it was negative or didn't fit in the narrative. The narrative is becoming increasingly important as years go on. And that article then is subject to curation, as we've been hearing about. And this involves steps of finding articles to look at, finding entities in the article, making assertions that link entities to other, by relations to other entities from ontologies, so that curation process involves extensive use of ontology and control vocabularies, and also filters of the data model for that resource. So there can be really important information that just isn't modeled in the knowledge base, so it gets filtered out, or the ontology isn't perfect. So it ends up with a subset of that information in the knowledge base. Okay, so this works, and those stars on the right, those objects in the knowledge base, are really useful for researchers to rapidly look at information about genes, cells, and so forth, molecules, but also to integrate that information. And it's computable because it's structured information. Okay, so the way I look at it is that there's levels of literature curation efficiency. So initially, 20, 30 years ago, there was expert curation, and that's very common, except if it's all text mining based. The software got better, I don't know, 15 years ago, where there were web-based tools, there's autocompletes for using the control vocabularies, drop-down menus, and the user interfaces for the curators just made a lot more things a lot more efficient. That's in common use. Entity recognition of bioentities also in common use, as we've been hearing in the meeting. And then it gets more elaborate, which is what I want to focus on and how we can move forward. So if you think about author curation post-publication, well, there are a number of systems. I think POM-based, the Saccharomyces POMB database led by Val Wood is one of the leaders in that, of getting the authors to add annotations and list assertions from their paper afterwards. They get very good turnout. It's a very tight-knit community, and from really stemming from a few labs, and that's probably why it works. But they've learned a lot about how to do that. So we've done that at Wormbase, and Chris Grove has led the effort, and so we send out emails to request published papers, the authors of published papers, to add information about, let's just say, a single type of information. Does this variant, this allele A, have a phenotype? And maybe a little more information about that. And our experience is that it works. It's a more efficient use of curator time doing the quality control and developing the whole process, but not highly leveraged, because of only about 20% of the authors respond, and the papers are only partly curated. But if you track that, at least we do get information. So there's 800 distinct authors as curators, led to 10,000 assertions, and there's more than 1,000 papers curated. I think the most really exciting thing about these data is that once people realize they could curate, then they actually send in information and self-curate without being asked. And it's about a quarter of the information. Okay, so that works, but as I think, as we can realize from this, and the fact that the idea has been around for a lot of time, it's not going to float the boat. Okay, another aspect that I think is now more common, so it's, I would say, few but increasing, is using text mining with curator review, so that you're getting around the inaccuracies of text mining by having human experts look at it. And I just want to kind of highlight, because it came up in the conversation yesterday, about how very simple method gives good leverage. So with Texpresso, which was, say, almost 20 years ago now, what it does is it takes single sentences or text spans and marks them up with categories of terms from control vocabularies or shallow ontologies, or even principal deep ontologies. So it finds genes by entity recognition, for example, words that mean regulation, life stages, bits of anatomy. This is C. elegans, so it has, kind of vol F as a piece of anatomy. And that allows really good characterization at a document or sentence level to retrieve sentences of interest. And I think one good use of that was semi-automatic curation for gene ontology cell components, and that Lynette discussed this morning in one of the biocreative tasks that this was successful. And the idea is that if you take sentences that have a cell component, like nucleus, A bands, basolateral, and apical membranes, you take a verb that has that is typically used to associate proteins with a cell component, like antibodies, protein location levels, or other words, or these are like stained, found, expressed, and appears. And you also have a protein. These are in capital letters, bold. You can then identify sentences that are relevant. You then can, on a form, I'll show you examples later, but you've seen them in this meeting, where you pull out the protein, where it is, and maybe something about the method, and let the curator say yes, no, or change something. And in practice, this is actually able to find new cell components, because it might say protein A is localized to the nucleus, the cell membrane, and the blah, blah, blah. And you know that probably tells you that extra blah, blah, blah is a cell component. Okay, so that text mining with curator reviews seems to work well. Okay, so the next thing that we've been doing the last couple of years is trying text mining with author review. So the idea is that the authors are busy and impatient, and you want to make it as easy as possible for them to give back information. Now, the first task we did was for document classification, where we'd successfully been using machine learning methods like support vector machine 10 years ago, and then various types of neural networks to identify papers and associate them with different types of data. And then we ask the authors, is this correct? And that helps create a virtuous cycle of training. So this is now, we're engineering the whole thing from scratch to make it more efficient. And this is this acknowledge system that I mentioned earlier. So the paper's published, it's processed. We prefer to get XML, but a lot of it is PDF to text, done on the fly. The paper's analyzed for data types using machine learning approaches. You identify entities, identify the corresponding author, and then you send the form to the author. And the form, which should be very easy to use and pretty, and are getting better, list the types of entities are there, and the authors then ask, is this true, and should they remove or add other genes, for example. And then there's various types of information, for example, phenotype and function, and the authors ask, is there phenotype information in this paper? And if so, are you willing, it's not required, to add that information into a form? So, we're getting about, I'd say, 50% response on this at the document classification level. And we need to make it very easy for authors to add information. Okay. So the next, the next level is, well, some of the authors have moved on to other careers, if they're students and trainees, or by the time the paper comes out, it's been so long, some authors don't want to think about it, they've forgotten about it. It might even be years since those experiments were done. So we'd really like to capture the author's attention before publication. Now, okay, so here, let me, again, coming back to the data lifecycle, as I mentioned before, the papers is curated after the fact, and it's lossy. What we'd really like is to have the authors involved earlier. So during the preparation of the article, that the authors are actually interacting with the ontology and the data model filters and expressing their assertions in a structured manner. And then the article would be, they'd finish the article, and in parallel, you get information to flow to the knowledge base, right? And this makes the most sense, but it's also the hardest to implement. So one problem is that, as many of us probably, as some people have mentioned, it's also in a lot of our experiences, is that if you interact with journals, it's very hard for them to change their workflow. Even so simple things like interacting with a journal website on a revised paper versus the first submission, it's still a pretty kludgy because the software isn't re-engineered to make it easy in later stages. So if you asked those workflows to include some curation step, it just doesn't happen. Okay. So this is one aspect that we also thought about. Other problems with current scholar communication. The main thing is data knowledge doesn't flow into knowledge bases, and also, as we won't discuss here, articles are too complicated to read, write, and review to ensure reproducibility. So because of that, we decided to try to explore what would it be like if curators control the publication. So we started with this micro-publication concept that's published by Caltech Library and at www.micropublication.org. And what the idea is to have very short articles that allow, initially a lot of information that wouldn't be published to be published. So we're working in a space where it's really just it's not going to get published, so the authors are willing to play along with us. Okay, so with micropublication, what we're working on is having the authors do this step, but it's very simple, and do the interpretation writing and use the ontology and data model filters. So this is curator control of scholar communication. Okay. So what we have developing is we have an article, we have an entity linker that allows us to find every entity, and this is controlled down to, in this case, alleles, methods, cell components, et cetera. We've heard a lot about that. There's great work going on. And the idea here is just to have it at the time of publication. One thing that this does is allow biological copy editing. So, we talk about identifiers being mislabeled. Well, wouldn't it be great if the author got feedback about their identifiers and saying, is this really a new gene or is it a typo? And when we've done this as part of the publication process with the Genetic Society of America journals, Genetics and G3, there was a pipeline that was feeding into various of the model organism databases, including Wormbase and SGD and Flybase, where the curators were having access to the nomenclature in papers, and they're finding lots of mistakes and cleaning it up. And we think that's would be a good approach. Okay, and then we have this graphical curation tool, which is trying to make it really simple for a curator or in this case, author to structure the information in the paper. So here's a tab for gene expression and saying, okay, here's a gene, UNC104, it's expressed in the ventral nerve cord. There's nothing about the life stage, and it's localized in the microtubule using some kind of reporter gene, presumably translational. And they can then create an annotation. Here's a same thing for a different kind of gene expression model where you have a gene, it's in a tissue at a certain life stage, and it's using a different method. And you can visualize the simple database record where you have an ID, a gene, these terms for ontologies, and a timestamp. So this is going to allow the during publication, pre-publication process. So what that does is it's using text mining to allow the authors to review, and it's pre-publication. So we think that's going to be, the best state of the art. Now the future really would be, to capture assertions and write the paper for you, but that's kind of a fantasy. And that's what, so to sum up, to how to scale, I think we got to combine more expert curators, really good user interfaces with other software, and engage authors and journals in a deep way in the process. So thank you for your attention.
Great, thanks very much, Paul. We have time for one or two questions if anyone would like to ask.
Q&A.
Okay, if there are any, please feel free to add them to the Q&A, address them to Paul, and we'll get those answered. So let's move on. Our next speaker is Dr. Cynthia Smith. Dr. Smith is a research scientist at the Jackson Laboratory. She joined the mouse genome informatics team 22 years ago and currently leads the gene functional annotation and phenotypes and disease model annotation groups there. She is the PI for the CRE portal resource reporting recombinase activity in mice and the International Mouse Strain resource, a searchable online database catalog of mouse stocks available worldwide. Dr. Smith also founded the mammalian phenotype ontology, which is used to describe mutant phenotypes at MGI, the rat genome database, and other resources. And today she will tell us about advances in literature curation at Mouse Genome Informatics.
Thanks, Kimberly, and thank you for the invitation to talk today. I'd like to talk to you about changes that we've made recently in our literature curation pipelines to support literature annotation in the face of increasing amounts of data, as well as reduced numbers of curators at our resource. So data acquisition and integration at MGI, we collect information on the laboratory mouse, including gene function information, phenotypes, mutant alleles, and models of human disease, gene expression, strains, mouse models of human cancer, and many other types of data. These data arrive at our database with over 50 data loads from external resources, literature curation, which will be the focus of my talk today, as well as submissions from authors. We use a myriad of data standards and ontologies, including gene ontology, mammalian phenotype ontology, and other mouse anatomy ontologies to classify and integrate all of this mouse, as well as relations to human data. All of our data is available free of charge from our website, APIs, downloadable reports, and batch query forms, as well as the web interface. We also export these to a myriad of other resources, such as various NCBI databases, as well as the Alliance of Genome Resources. So literature curation at MGI, PubMed is adding an increasing number of new mouse papers per year. If you look at this graph, if you search PubMed with just the term mice, over the years you see a very high increase in the number of papers that are returned using that term. However, at MGI, the number of papers that we have been curating over the years has remained relatively consistent, certainly over the past 10 years. The 2021 value is a little bit down because we haven't finished incorporating all of the 2021 papers yet, because we have a one-year embargo on access to a number of journals. But we see this every year, and I expect that that will rise up to a consistent level as we go through 2022. So MGI currently adds about 12 to 14,000 relevant papers per year for curation, and discards an increasing number of papers per year. So the question is, why is that, and how can we automate this process so that curators aren't spending all of their time throwing away papers? And this is very time consuming, so we really wanted to automate this process. So first, we obtain reference PDFs and create MGI records. Prior to 2015, this had been a manual process, but we now download PDFs in bulk from PubMed Central with 77 journal titles, and Science Direct for 45 titles. PDFs from other selected journals are still manually downloaded, but we have a lot of administrative assistants to help us in that process, as well as some of the curators who have favorite journals. We then take these PDFs. They must contain the word mice somewhere in the text to be loaded to MGI. And we extract the text out of the PDFs. I'll get to that more in a moment. The DOI or the PubMed ID is extracted, and the full citation information is retrieved from PubMed to create a reference record in MGI that's associated with the PDF and the extracted text. So that's now automated. We also then further analyze the text in the PDF by sectioning the text within the article. We then pull the text from the body, the references, the figure legends. Some journals have something called a STAR method section, which lists reagents used in the paper, as well as a supplemental data section whenever that's available. The text is extracted and split, and then the splitter further searches the fields for the term mice. If it's not found in the body, the supplemental STAR methods or author figure legends, then the record is set to discard, and the curators never look at it, and a tag is added, mice and references section only. This eliminates about 5% of our loaded papers. So we then wanted to further incorporate machine learning into the literature triage for relevance. So after the mice papers are loaded into the database, we want to predict whether we want to keep or discard these papers, just the MGI relevance decision. The motivation was that it would ease time to evaluate all of these papers. Initially, we were discarding 15,000 papers a year. That's increased over time. And it certainly would allow more curator time devoted to data entry. This particular task was costing 3 to 5 curator days per month just to select the literature from the corpus. So a robust machine learning classifier was developed and trained on our already curated articles at MGI. In our implementation, we used a gradient boosting decision tree model with some text preprocessing and vectorizing. And that one statement is kind of a disservice to the amount of evaluation and that went into deciding on this particular method for machine learning. So our relevance classifier was trained on our keep and discard set from about 80,000 PDFs in the MGI corpus that spanned the publication dates from 2017 to 2019. And it was important to use the full text because we found that the relevance classifier did not perform well on just titles and abstracts. And the full text test set was just under 23,000 PDFs that we collected in 2020. When we looked at the results after a lot of tweaking, we found that the precision and recall were quite good, as well as the negative value. So we were confident that this step is eliminating about 60% of the papers that we were loading without any curator intervention or evaluation. Now, there are some degree of false negatives and false positives. The curators can change that discard or keep status manually when they run across these. And we are planning a retraining of the classifier soon based on our additional curation over the past couple of years. We also track the relevance history. We store the predicted and curated relevance assignments initially when the paper comes into the database. It has a not specified relevance after the relevance classifier has processed this paper. It's processed as either keep or discard with varying degrees of confidence. A high confidence paper will be fast-tracked for curation. The date that it was classified in the version of the classifier is also included. We also store when a curator has actually looked at this paper and verified that we want to keep that paper. And any time a paper is curated, the relevance history is automatically updated to reflect that a curator has validated this choice by the relevance classifier. And ultimately, we will need this relevance tracking for classifier retraining. Literature curation at Mouse Genome Informatics is split among different workflow groups that evaluate and curate selected data types. So for example, the gene expression group looks for expression in during development. The gene function group is looking for Go functional annotations as well as subcellular localization. The alleles and phenotypes and disease models groups looks for new mutations as well as new phenotypes on existing mutations and human disease model information. Splitting the curation among these groups allows curators to select the most robust or relevant papers for each data type, given the amount of volume of papers that we deal with. And so we wanted to come up with a way of now taking the papers that are that were deemed relevant by our classifier and put those through a secondary routing process for each one of these curation areas. So we go back to that extracted text from the PDFs, and we search it for the frequency of relevant keywords for each one of these curation areas. The secondary routing only considers the relevance status of keep and not discard, and uses the group specific keyword as well as exclude words for those particular areas. And then this process sets the group status to either rooted or not rooted, as the case may be. So we analyzed in our test set 9,322 papers from January to August 2019 that had been fully indexed or curated during that period of time by our curators. And we compared the results of the automated secondary routing to what the curators had entered in our database. And for the alleles and phenotypes group, the recall and precision was quite good. Of those 9,000 papers, almost 8,000 matched the criteria that we specified in search terms that we derived from existing papers, and several, and these terms were from existing AP reports, allele collection names, and some relevant ID prefixes. For QTL, this was a little bit different. The recall was nearly 100% for one single search word, but the precision was not very good. But for the number of papers that were retrieved by this simple search, we decided that we could live with that we could screen through those to find the papers that we really wanted to curate. Similar for gene expression database, initially the recall was very good, but the precision was not, and some adjustments were made to the keywords that were used in this secondary routing. And similarly for the tumor database, the recall was good, but again, the precision was not great. This was further refined in subsequent versions of the secondary routing. And as data changes, as new technologies come along, we can change these criteria quite easily to accommodate those and improve our secondary routing. Another part of the secondary routing for Go in the gene function group involves gene indexing. For each one of these papers, we actually go to NCBI and collect information out of the gene to PubMed file that they produce on their website or in their downloads. Out of this, we select mouse taxon gene associations to papers, and then we load those into MGI and associate those with the papers. This is run weekly. It's a complete drop and reload process. So if the PubMed has been imported, if the PubMed ID has been imported to MGI, marked as keep, and it's associated with not too many gene IDs, we then store that gene ID to MGI reference record in MGI. So each data area has additional literature reports that exist that assist in tertiary prioritization for curation. So for example, our gene function reports will list papers that are now associated with genes that have no experimental annotations in MGI or are associated with genes with underrepresentation. So for example, if a gene has 3 papers curated to it for gene annotation, but there exist 77 papers with associations to that particular gene, we may have a second look at that paper. So the PubMed index, the PubMed to gene indexing allows us to automatically generate these reports and list high priority papers for gene function annotation. Another example is with allele curator reports from rooted papers are generated with snippets of text surrounding those keywords that we chose for that secondary rooting and include the associated genes, which will help us prioritize papers that have phenotype information for genes that we do not have any information for. And that can look something like this. This particular report is to find papers that are highly likely to have new alleles in them. We have the papers that are rooted or chosen for our group. They're not in a discard pile. And within that paper, they have one of these terms. Each one of the results is listed with a snippet of text that gives us an idea of whether this is a paper that we really should look at or whether it's referring to a previous study. It's also associated with the mouse gene IDs, which we can use to identify papers that may be in alleles in novel genes that we don't have any information, other phenotype or allele or disease model information for.
About 2 minutes, Cynthia.
Okay, well, I'm just wrapping up.
Perfect.
So in summary, as a beginning into our foray into natural language processing, we've initially developed a robust pipeline to load and screen for all of the relevant literature for expert curation of multiple data types for mouse in MGI. The relevance classifier initially has been eliminating over 60% of those loaded papers automatically so that expert curators can spend more time on complex curation tasks. The automatic secondary routing has been in place for almost a year now and selects high priority papers for specific data types without manual curator evaluation using keywords and automatic gene to paper indexing. We're constantly reevaluating these secondary processes, especially given the light that we're discarding more and more papers just to make sure that we're not eliminating papers that we might want to curate. And in addition, we will continue to participate in natural language processing projects to further extract data from papers at MGI and the Alliance of Genome Resources. So it would be interesting to now look at named entity recognition methods to pull into our pipeline to assist with the data curation. And so interestingly, we're still only pulling in the same, even with all of the relevance classifiers and with the secondary routing, we're still only pulling in as relevant the same number of papers that we have over time. So it would be interesting to analyze all of the discards to see why that is. Empirically, I can tell you that a number of a large chunk of those papers talk about mouse phenotypes from previous studies in the context of human disease papers. So there's no novel data in those papers to curate. We're also seeing things like drug studies in mice, in wild-type mice, which is beyond the scope of our annotation, unless they're within human disease models, which we would take. And so we're not really, since this is all automated now, we're not really listing the reason for why we reject some of those papers. But maybe if we look at some of the false negatives or false positives and true negatives, we can get some more information on that. But that remains to be determined. So I'll stop there and acknowledge that the majority of this work was led by Jim Caden, one of our software team members who is now semi-retired, along with Richard Baldarelli and the rest of the software engineering team, with significant input from the MGI curators. And this work was funded by the NHGRI grant to the mouse genome database part of the MGI consortium. And I thank you for your time.
Great, thank you very much, Cynthia. I think discarding 15,000 papers is amazing for those of us, right, who curate probably 1/10 of that. We have one question in the Q&A. Mike Bluestone asks, with so much reliance on automated methods, how do you guard against false negatives?
So we do constantly reevaluate all of our methods to determine what we are collecting. Certainly we need to rerun the relevance classifier. We need to reestablish that. But I think that ultimately we need to actually look at some of those papers and determine why they end up in that pile and adjust our methods accordingly. It's not a perfect process, and neither is human curation. But we do have the ability to go back and pull those papers out and look at them if we need to. So.
Okay, great. Well, thank you very much, Cynthia. Let's move along. Our next speaker is Dr. Ann Kwitek, an associate professor of physiology and medicine at the Medical College of Wisconsin and PI of the rat genome database. Dr. Kwitek is an animal and human geneticist whose research revolves around dissecting the genetic components of complex disease with an emphasis on hypertension, diabetes, and metabolic syndrome. In addition to animal and molecular genetics research, Dr. Kwitek has worked on developing knowledge bases and other resources for the laboratory rat and the scientific community. And today, Dr. Kwitek will tell us about increasing curation efficiency through automation-assisted manual curation.
All right, well, thank you so much for the invitation to speak today. And I have to mention that this talk was supposed to be done in 2020 by Dr. Mary Shimiyama, who was the head of the RGD at that time and unfortunately passed away. So I am going to do my best to represent, and I have been representing the group for the last few years. Okay, I'm having trouble. Okay, so the Rat Genome Database was established in 1999 and with the goal to curate genetic information for the laboratory rat. And it's really evolved to a comprehensive source of rat genetic, genomic, phenotypic, and disease-related data. And so, unlike some of the other organism databases, we made the decision to take a disease-focused approach to literature curation. And we use an automation-assisted manual curation, which I'm going to talk about today, and developing and using robust data standards and ontologies. And as a result, we've generated disease portals to access the data and tools related to so far 15 diseases and across 10 different species. And we've always been focused on not only curating rat data, but really integrating that data across to human disease and also other human disease models, such as mouse and several other species. So the manual curation is comprehensive and diverse. Like I said, it's disease and gene focused. And so we select a specific disease focus and then really go in and dive deep into the curation of that particular disease area. And so, excuse me. Sorry about that. And so we will take a disease area and then we used cross-species curation focused on the laboratory rat, but also including human disease and phenotype data, and then some limited curation of other species that are represented in RGD. We look at multiple data types and ontologies, so including genes, gene alleles, QTLs, and strains are all manually curated. And then disease and phenotype ontologies, gene ontology, pathway ontology, gene chemical interactions, and also we do a fair amount of quantitative phenotype measurements rather than just qualitative annotations. So here's the challenge. If you do a search of PubMed for rat, it returns about 30,000 papers per year since 1984. And RGD has always been a relatively small curation shop. So we have a relatively small number of curators. So we really had to be able to address how to look at these this corpus of papers and be able to curate out of those. And so really the first enhancement of efficiency was to change from Excel spreadsheet curation into a very nice in-house a suite of software, curation software, and that was done in like you can see in 2006. And when you look at the curve of the cumulative count of annotations, you can see that greatly enhanced our efficiency. So we could get a lot more annotations, and it also helped to decrease errors in getting information into the database. So the curation itself is sort of what we call a bucketing system, and it's really a one-stop shop to curate multiple objects, multiple ontologies, and references simultaneously. So unlike like what Dr. Smith was saying, we have one curator who really pulls all of the information out of a single paper that has a disease and gene focus. And so we're able to add objects, ontology terms, and references all in one fell swoop and bucket them into the selection for what annotations would be added for that particular gene and disease or phenotype. And we found that it helps with less copy and paste and fewer errors. And it allows you to mix and match the selection, and then the curator can review those annotations, and then the information is entered directly into the database. So the curators are putting information directly into the database, which helps reduce lag time. So there have been several software improvements that have been done over the years since that first round of curation software. One of them is to be able to search for one gene, for instance, and add the whole ortholog group into the bucket if we're going to try to do disease and pathway annotation, for instance, across rat and human. Another improvement was to be able to automatically assign an ontology type based upon the term selected, and I'll show you that in a moment. Also, there were integrated QCs, qualitative control of qualifiers, and evidence codes based on the type of object and ontology that was being selected. Finally, automatic addition of inferred by sequence orthology annotations was proposed, can be proposed to the curator, and then when it's appropriate, then they can accept those into the annotations. And a final thing that really helped out streamlining the curation process was to integrate our RGD's ontology browser, which you can see a little picture of at the bottom of the slide here, into the curation software itself. And so then if a person is looking for a specific term and then realizes they need to either focus in or become more broad, they can either go into a child term and select that, or back off into one of the parent terms and select that. And that also just helps from having to go elsewhere into a tree to be able to select terms. So another major enhancement that we implemented was utilizing what we call Ontomate, and this is a text mining software. And really, we did this because, as you remember, I told you we do curation by a disease and gene focus, rather than looking at the entire corpus and then pulling things out that might be relevant. We start in with an idea of what we want first. So the biggest bottlenecks that we found were finding those articles that were appropriate for curation and then extracting that information from those selected articles. And so Ontomate was developed as a text mining tool really to aid the curation, not to replace it, but to really help the curators focus in on and find what they want to be able to use for curation. So this tool is fully integrated into the curation software, but we've also made a web version of this. So it's on our website that anybody can go in and utilize the search engine from Ontomate. But within the curation software itself, you can see here we've selected the core objects, which are as the gene CFTR from rat, mouse, and human, and those are added to our bucket. And then the ontology term in this case is cystic fibrosis, and that's put into the bucket. And then there's the ability to search and Ontomate to be able to use the available object metadata, such as the gene name, any synonyms, the symbols, and etc. And then also the structure of the ontologies that were selected to be able to identify relevant abstracts from PubMed. So this is a kind of a busy slide, but I'll try to take you through hopefully without having to advance a slide. And so here you can see if you search for CFTR and cystic fibrosis, you very quickly get about 8,500 results found in PubMed. These are sorted by a relevance, which is scored by being most likely to be curatable, so you don't have to scroll through 8,500 or 8,600 different papers. It also gives facets here to be able to easily narrow your results either by a year or by organism, gene, mutations, or diseases. And so, for instance, if I only want to see, for instance, here, rat articles related to cystic fibrosis and CFTR, I can just select and filter on that. Right away, you can see that the gene and the disease term matches are highlighted into the results, and the abstracts are incorporated into those search results. along with links to NCBI, the PubMed, and also PMC for the full text. Now, if you go through and open up the abstract, you can see that that is also tagged with the ontology terms, the gene symbols, and any of the organism taxonomy names from various sources. If you mouse over a term in these lists, so this is down below here are all of the various ontologies that have been tagged out of or possibly tagged out of this this particular abstract. If you highlight or if you mouse over, you'll see where it is in the text. And if you click on one of these terms, so for instance here, cystic fibrosis, it's actually part of the tagging process that it's cut off, but you'll actually go directly to the embedded ontology browser, so that again you can either select that particular term, its children or its parent type, and then you click on this little load icon here, and it loads that ontology term into the annotation. So when we looked at this, so Ontomate was integrated into the curation software in 2013, and I think it's kind of clear to see that it was a benefit to our curation efficiency, so that we were able to essentially double the amount of annotations per curator. So this is average annual annotations per curator, and you can see very nice increase in the number of annotations that were possible by just integrating on this search engine into the curation software. Obviously, we also do a lot of automated curation pipelines to import data from other resources who have done their wonderful curation, and so we bring in several pipelines to complement the manual curation and really prevent the duplication of efforts. And so these automated pipelines have been a lifesaver, because we can bring in many different types of data, such as, I'm not going to read all of them, but gene, transcript, and protein data from their various sources. We bring in orthology data from HCOP and now from the Alliance of Genome Resources. We're bringing in variants, whole genome variants from ClinVar and from EVA for selected species. And the various ontology annotations from non-rat species from many different expert resources, gene chemical interaction data, protein interaction data, and microRNA target data. So this has also helped to complement the manual curation and make sure that we can be comprehensive in the amount of information we represent. We've developed some QC pipelines within RGD's existing data sources to check and correct data in the database. So for instance, if a symbol of a parent gene would change, the pipeline automatically identifies those changes and changes the corresponding allele and strain symbols to match those. Another example is, for instance, if a rat gene symbol doesn't match the human and mouse orthologs, that information is fed into a different curation tool that allows a curator to review the orthologous gene symbols and where needed, make changes. And right now we're going through a lot of those. So that's a very handy tool. And there's several more that I'm not going to go into right now. Finally, we do encourage user-submitted data into RGD, and for instance, for strains, particularly strains and alleles. And so we encourage investigators to submit their alleles and strains pre-publication, so then they can have a reference identifier to put into their publication, and so that really helps with with the nomenclature issues that revolve around strains and alleles. And recently, we've sort of streamlined this process by providing a more user-friendly form for a user to submit. And of course, we'll help with assigning the nomenclature, the symbols. But by giving them this form, then they have a way to provide this data in a standardized way. And then that gets migrated over to the curation aspect, where the curators can either create an allele or create a strain, and the information that was provided by the user is automatically populated. And then that strain or allele can be made public either immediately if the user wants that, or upon publication. So we can hold off until they tell us that we can go forward. So I didn't talk a whole lot about the quantitative phenotype curation, but this is a really complex curation process where we're actually bringing in quantitative measures and also associating them with a minimum of at least 5 different ontology terms. And we're really the curation software could be improved to increase the efficiency of this. And one thing that we need to do is better link the quantitative phenotype curation with the qualitative phenotype curation. And also, we really like to expand Ontomate to help extract quantitative values and terms. So in previous talks, in the previous session that we're talking about, being able to tag supplemental material or figure legends or figures themselves, because we can't take information off of a bar graph because it isn't an actual quantitative value. So having the ability to improve and expand on Ontomate to help extract those values and terms would be really helpful. And of course, being able, the elephant in the room for Ontomate is that so far it's been limited to tagging abstracts, and so a main goal is to really to be able to utilize full paper tagging with Ontomate, and we look forward to working with the Alliance of Genome Resources with they're building a really nice corpus so that we can potentially test full tagging. But the advantage of that is because it's comprehensive and it has all the ontologies, it goes across species and so forth. And another thing that we'd like to learn from, as Dr. Sternberg was talking about, is engaging community in our curation processes. So we've done a pretty good job with strains and alleles, but it would be really, we're looking forward to continuing to work with the group at the Alliance to include potentially author first pass, where as Dr. Sternberg talked about, tagging with the provided standardized vocabulary terms. We'd be also interested in learning more about improved text mining algorithms. And so with that, I'd like to acknowledge the RGD team. We have an expert group of curators and developers, and we're funded by the National Heart, Lung, and Blood Institute, and we're also part of the Alliance of Genome Resources. And with that, I'd like to thank you and take any questions.
Thank you very much, Anne. That was really comprehensive. We have time for, there are 2 questions in the chat. So from Kristen Markey at the EPA. First was, while you're presenting Ontomate, if Ontomate is searching by ontologies, and say, for example, the query was just Rattus species, does Ontomate use an algorithm to pull in other things like UMLS or another semantic resource to generate the query? In other words, does it go beyond the mesh terms?
Yes, it does. So it uses mesh terms, it uses the titles, the abstracts, the author lists, the citations, and then like I said, I'm not an expert at this, so if we're talking about really the technical aspects of how these algorithms have been applied, I'm going to have my group to go into the Q&A into the to do this offline, or after I'm done talking. But yes, it's not just by mesh terms.
Okay, and the second question, is your curation software available for use?
So theoretically, we would be happy to make it available for use. It's just it's really plugged into our database. So if somebody's interested, please contact me and we can discuss how we might be able to do that.
Okay, great. Thank you very much, Anne.
Thank you.
Okay, and so our final speaker for this session is Dr. Mike Cherry from Stanford University. Dr. Cherry has been creating model organism databases for over 30 years. He is the PI of the Saccharomyces genome database and one of the PIs of the Gene Ontology Consortium and part of the Alliance of Genome Resources. His lab is funded as the Data Coordination Center for large NHGRI epigenomics consortia and also leads curation for the Chan Zuckerberg Institute's Cell by Gene resource part of the Human Cell Atlas effort. And today, Dr. Cherry will be presenting on a philosophy and its implementation of these requirements for gold standard data and metadata.
Thank you, Kimberly. Good morning. At least it's still morning for some of us, and should be able to see my slides now. So it's no surprise that I completely agree with Paul Sternberg on the progress and needs for biocuration tools. Of course, this needs more tools, needs biocurators, needs the community involvement and such. I want to say up front, excuse my opinions here. I strongly feel strongly about some of these things, and so they'll pop out as I would think they need to be. I'm going to deviate a little bit from the others in this section on mods and talk a little bit about the data coordination centers that we've created in the lab. This mix of projects between literature curation and data curation has actually had an interesting impact on sort of everybody. And so while my title is philosophy and implementation, it's really sort of more like summary and problems and challenges, I guess you could say, within these projects. And so I'll talk a little bit about the two main projects in the group, the Saccharomyces genome database and the ENCODE data coordination center. And so you're seeing my ID there, the lab website, and my funding acknowledgements. Okay, so my lab is interested in this mix of projects that span the range of biocuration and data wrangling. On the left, you'll see SGD is a model organism database, the Alliance of Genome Resources, which is a consortium of large mods, the Gene Ontology Consortium that continues to enhance the Go ontology, creates function annotations, and develops tools. I use the business title Biocuration Scientist instead of Curator. I think Biocuration Scientist is a more appropriate term, and it actually gets to the recognition that these highly trained, educated scientists need, rather than just Curator, which I think sometimes people don't understand what that means, and it sort of belittles the importance that these individuals have. And so just like on ontology, when you consider a term, you need to look at the definition of that term. Curation is an interesting one because it's often used in many ways these days. For example, you can curate a playlist, you might curate your fall fashion selections. Clearly this is not what we're talking about today. But people will know of things like museum curators, which is a hard task, and it involves a lot of knowledge there. So biocuration is thus my preferred topic, and biocuration scientist comments to the people. So the use of Curator also, I think, has an impact on researchers. I think some researchers think that curation is easy. They don't quite understand why we need all this metadata, why it's hard to get metadata right using ontologies and these sort of things. And I would say, surprisingly, even some directors get confused about what's involved with biocuration, and it would be sort of really nice that they would, if nothing else, look at the last session and sort of understand why humans are really necessary, what's possible with computers today, where we hope they'll go. Okay, so the projects on the right here, the ENCODE, the IGVF, which is impacts of genetic variation on function, quite a mouthful. IGVF is one most people say because they can't remember the full name. And then this lattice effort here, which is part of the HCA data coordination as such. Okay, so these are all large, ENCODE being the largest consortium here. ENCODE is 23 years old, and we're in the last year of data generation. So the work that we've put in up to this time in creating this very standardized metadata is so important to allow this data to live on. IGVF is really just starting. There's not any experiments that have been submitted yet, but the experiments would be very complicated, fewer in number. And the complication comes to the fact that it's going to be a lot of massive CRISPR screens based on single cell readouts and such. As well, the assays are not really standardized yet. So we're still trying to get a grasp on what it is that we actually need to do as far as what the metadata models will actually be. And then Lattice here is leading the curation of the cell by gene open resource that the philanthropic organization Chan Zuckerberg has funding. They fund a lot of single cell research, and then we're aiding with that, as well as putting in some community efforts as well. That's actually fun because it brings us into connection with other big consortium projects, such as the HubMap, and working with other large projects, such as the data coordination platform, part of the HCA. And by the way, I mean, HubMap is really doing an amazing job with ontology development. They probably would have been an interesting one to add to this meeting as well. Okay, so these three projects do a lot of data curation. And for them, I use the term data wrangler. They have many of the same skills as biocuration scientists, but they require a bit more expertise on primary data and its analysis. Okay. And I must say, it's really been nice that the NHGRI has been funding ENCODE and designing ENCODE and IGVF with a lot of specific sharing in mind. So these are open science, open data sharing, open reagent sharing, open source software, open access in general, shared robust software tools, and shared standards. So they're really, everything is publicly distributed, publicly available, transparent. Okay, and I think that's really important for these. And it understands the funding of these data coordination centers helps define that we help define the metadata, allows these data to be rich and complete, and that the data resources then is much more valuable to the community. With these large consortium, we strive to regularize, harmonize, and I guess to use Melissa's term, homogenize the metadata so that for the whole data corpus. This is a lot of work by these data wranglers and actually software developers as well. Okay. For the projects on the right, we get the data right off the sequencing machines. Before we receive the data files, we work with researchers to define the metadata for the experiments. Also, the results cannot be released until the metadata and data passes QC. So there is a large incentive to create complete metadata that meets the community standards that have been defined. Okay. This all happens before the analysis, the definition of the metadata. It all happens before the analysis is started, certainly before it goes onto the machines, and much before the paper is actually written. And so we really get this rich metadata on the analysis products, software used, and even the options used for that software. So the projects on the left, the SGD, the Reliance, and the Go, these are literature biocuration projects. The authors, do not provide any metadata, and at minimum, they give what tiny bit the publishers require, IDs for sequences. Thus, metadata is basically non-existent in a downloadable form, and we often get little more than We get a little more when we contact the authors. You know, students have moved on, the lab notebooks have been put away, new projects have started, you know, et cetera, et cetera. Okay, and it's obviously, you know, many times it's 6 months or more, but that the work was actually done before we actually see the paper. There have been discussions for a couple decades about making structured papers. I know it's mentioned a little bit earlier, and it's sort of interesting that this hasn't happened. You know, there've been several good ideas with time, some software tools have been written, but you know, it's why hasn't it happened? Well, to me, you know, like so many things in the world, it comes down to money. And really, until the NIH says you have to provide your data in some sort of computable form connected with a publication, you know, it won't happen, right? You know, the funding can drive these kind of things, funding awards. Okay. Okay. So then here, so talking about the ENCODE, so it's essential that we capture complete and rich metadata using standards. Over the years, we've collected roughly 20,000 experiments, submitted from hundreds of different assays. Several 100,000 files have been included in this, primary data files, analysis files, and such. So in total, we're distributing roughly 1.2 petabytes. It's completely available for use and download in and at the cloud. All the metadata is stored in a separate database and an object-like database, and all this is available for download, searchable by our web portal and such. Okay. So a bit more focus here. So some of the entities that are shown here are described in our metadata. We use standard ontologies. You know, we try not to develop an ontology. That's sort of you know, we don't want our own standard. We want the community standards. And so we spend a lot of time working with others, enhancing their ontologies. And this really has a wonderful result, is that when we work with other data coordination groups and use the same ontologies and harmonize our metadata schema, we all are a bigger success because we can share our resources, processing pipelines, tools for creating the database. But more importantly, our users can actually query us and find the same tissues, the same cell lines, the same assay results, you know, because it's all the same. It's all the same ontologies. And so we have lots of QC checks that go into experimental data and assessing the experimental data and the metadata. One of these things is the provenance of the information. As illustrated here, so we have results of the analysis, so there's peak files. Right, so that's the information that might be plotted on the UCSC genome browser. This comes from alignments of the reads to a genome. Those reads are from a library, and the library is from the tissue, and the tissue is from a donor. So we need the metadata to allow us to trace back through all these things. For these alignments, there's also association with certain software tools, and as I was saying before, even the options used on those tools. So the results can be reproduced. Okay, so now then switching to the literature curation, this is a figure you saw earlier from Lynette Hirschman. She published it a bit ago. So I've just sort of re-annotated some of it for a yeast, an SGD specific thing. So SGD has annotations in the database from roughly 100,000 papers, full text, you know, when we can get it. Normally we can, and we also download all the supplemental data associated with the papers. And as noted by Cynthia Smith earlier, I'm pretty sure she mentioned this, is that the number of publications appropriate for us to curate is not growing exponentially. There's maybe a slight increase. It's often steady from year to year. I believe ENCODE has, Uniprot has published on this. This level of increase is not a problem for us. You know, we have and continue to be more efficient. What is a problem is the huge funding decreases that we've received. I mean, obviously we can do a lot less without the people to actually do the required steps, and those people are biocurators. Okay, so what this slide shows is some of the things that others have talked about tools for. You know, we can identify the bioentities, the biological objects, we can associate them together, these tools can, following by finding the evidence from the assays that describe this. Unfortunately, these software can give us, you know, triplets of information. And sometimes the paper is a little bit more complicated. The English describing this or the organization of the paper is just more complex. And so we still need biocurators to go in and deal with a lot of the information here. This helps prioritize things. It does assist us. You know, there's a question earlier, I think it's from Kim. about, have we actually done the work to figure out how much time saving this allows the curators? And like so many things in the world, when we save time on this task, that allows us to do more of another task. So it is certainly helpful, but by no means does it save us curators and curators' efforts, because there's just so much detail that we can do with curation itself, and we can get a much more rich result that goes in here. So the curators are the time limiting aspect. Okay, so that's sort of one challenge, building better tools and allowing us to look at these triplets of information and figure out what's best. I think Dr. Jensen mentioned that, there's a lot of false data. Cindy mentioned there's a lot of false data in papers. Curators right now have to go look at that and sort of see if something looks suspicious, and maybe read some more of the paper to figure that kind of stuff out. But in general, one of this gets to sort of this harder challenge that we have, and that is the review of a collection of annotations on a gene and understand how those connect with the gene's purpose in the cell. We have lots of annotations from many papers. They're all sort of different levels in the ontology. And so we need people to look at that and sort of get to the understanding, what's the essence of the work that's been done, what's the essence of what's been indicated by these assays. Oftentimes though, a function of a protein is not known, but of course, using the existing annotations, we can often say something, and that may lead to more research. Help people design an experiment. So it's by looking at resources such as ours and the other mods. Okay. So at present, the biocurators are involved with reviewing the collected information. They typically, read everything that's already on our web pages as they're reading a paper to determine if something is new, but also understanding where it enhances the existing information. And so, thus the term knowledge base is appropriate here, because it's not just these lists of annotations, is that we're synthesizing things together. I sometimes like to use the word synergize the information together. And of course, as others have said, we're dealing with English or some language here, and humans don't necessarily always communicate so clearly, at least as clearly as a computer would prefer. Okay, so here's a paper on foreign-mediated actin assembly. There's really this question here: is there a phenotype represented in this paper, described in this paper? Okay, it's yes. And so let's look further here. So we have an observable qualifier pair that's defined by this statement here. And so we have actin cytoskeleton morphology abnormal. Looking at the details, of course, was really where going past this, because I know others have talked about it. You can look at various passages in the paper and figure out snippets of the details for why this phenotype has been described, and really get at some of the detail of what really happened when you deleted HOF1 within these cells. And so we need to find these details to really make a complete knowledge base. Again, we're not just about saying it's abnormal. Well, why is it abnormal? Give us some details. So one important aspect of it, and others have mentioned this or alluded to it in their text processing, we need to figure out the strain background. Now, even in yeast, it's hard sometimes to get the strain background. They don't actually tell you which strain was used. Of course, it may be a complex mix of different strains. And sometimes it's even which yeast was used is a problem. So, these are sometimes not so easy for a computer to pick up. And then, of course, to make the complete information, we need to know about the alleles that were present. Okay, so it comes down to a sort of a neat set of details that have been integrated, that can be integrated and are integrated into our database. So the biocuration scientists are those that can do this. Without this information, individuals, some of this individual information might be lost, and except for a student or researcher going back and reading the paper. But once in the knowledge base, it's easily found. And of course, we make it available in a computable form. And that's the whole point, right? For a lot of this is we use ontologies and such so that a computer can act on these things and understand the relationships of these terms, whether it's a function ontology or an anatomy ontology or whatever. Okay, right. So knowledge bases, need to contain that knowledge. It's not just the data. We have knowledge bases we can get to understanding as well. And I think the graph goes that it finally get wisdom, and that's what we need the people to do and really come up with great inventions and understanding of health. So I would say it's difficult to understand why research dollars are being wasted, you might say, because all this data is being generated, it's being published in an inefficient way, and it requires humans to read it, right? And then we take it out and put it back into a database. And so clearly there's sort of something missing in this sort of process. And I think Paul mentioned some of those steps that can be made, but that's going to take a lot of time. Dr. Jensen mentioned similarly about the text processing. I think to quote him, he said it's at least 10 years before we can get like a human. I think I'd like to see that happen in 10 years, but I'm a little bit skeptical because it's a hard problem, right? So once the functions, interactions, associations have been described for yeast, of course, it has also been important not just for yeast researchers, but also other model organism researchers, anybody studying basic biology. And of course, it all leads to understanding human health. It is the National Institutes of Health, of course. Okay, so wrapping it up, here's the software developers and engineers, biocuration scientists, and data wranglers in the lab. This is really a fantastic group. They really make these projects a success. They figure out what to do. It's really through their efforts and insights and their dedication to this work, really to share everything. That takes more work to really make things robust. That takes more work. The literature curation teams here are less than half the size they were 5 years ago because of funding decreases. And so we've clearly had to be more efficient, but there's some things that we're not able to do. And we worry about these continued, I would say unwise decisions that have been made about our funding. Here, once again, is the lab's website. You can look at that to learn more about the projects and the people. There's links to all kinds of places in the publications from the lab as well. And so I thank you a lot.
Great. Thank you very much, Mike, for that talk. SGD has certainly been at the forefront of biocuration science, so it's great to have you here.